Hello. Yes. Y- Yes. Hello. Welcome to our today's session. My name is Delcy Shengoma from the National Institute for Medical Research. I will be sharing this session with my colleague, and it's my pleasure to present our first presenter, Professor Christian Happy, who happens to be my brother and a friend. Christian is a professor of molecular biology and genomics and the director of the World Bank funded African Center of Excellence for Genomics of Infectious Diseases in Redeemers University in Nigeria. Christian has lost say so because we are short of time. Christian, you will pardon me to end here, and given time, I'll give more about your bio, some of which I happen to know myself. So Christian, take the stage, and we are happy to get to hear from you.
Thank you, Deus, and thanks to the organizers of the Tanzanian and African Society of Human Genetics for this invitation. So, and I want to say that it's a pleasure for me to be here today and definitely presentation. And I will try as much as possible to be time or to narrate this story in a way that makes sense. I was told to present on genomics of Ebola, HIV, and COVID-19 in Africa. And though, as Deus has said, I had the World Bank African Center of Excellence for Genomic of Infectious Disease at Redeemers University. So we are a center we are at the WHO and Africa Union, reference laboratory for genomics and research and innovation, in Africa. And as such, we have a mentoring role and then also a quality assurance and supervisory role over the national and the regional labs across the 54 African countries. So today I will probably just go, and then as we talk about, we're in the era of pandemic, but then, on the slides is basically to show the position of Africa. And if you see, for instance, in this map, and then you look at all of these viruses, and these viruses that have pandemic potential, which includes HIV, Ebola, SARS-CoV-2 now, you realize that the majority of them are in Africa. And then the reason, why Africa, and then that really points to the fact that naturally, Africa should have been the world leader in terms of preferring solution to this to this disease or to these pathogens, that are pandemic potential across the globe. So across those, among those pandemic, you have Ebola, you have Lassa, you have HIV, you have COVID-19, although did not originate or emerge from Africa. But then this is really one clear evidence why, Africa should be the leader, in this area. So we really were very cognizant of this fact. And then as far back as 2010, we started thinking about how do we give Africa the prominence that it needs and the place that it deserves in a community of nations or in the globe, because Africa is endowed not only with mineral resources, but also with very extensive biodiversity. And as part of that biodiversity are viruses and pathogens. And naturally, Africa should be the world leaders, as I said before. So we thought about, how we can start bringing in genomics, in the early 2000s, in this space, simply because we realized, that genomic sequencing technologies have revolutionized the way healthcare research and healthcare and then research has been approached. And that has created a huge divide between Africa and the rest of the world. And these divides are not only financial, but also in terms of, benefit in enhancing science. And we thought that, embracing genomics and then start bringing that genomic revolution on the continent to address things that are very peculiar to Africa would have actually been very tremendous. And in the next few slides, we will see how we really started pioneering this on the African continent and how we're able to use that as a way to transform things like outbreak response to diseases. And I think to do that, we set up a platform, and then that platform, the African Center of Excellence for Genomic of Infectious Disease. And on this slide, you see, the African countries that are involved in this, and then also the partners, overseas partners, even though this has grown so much. So, and the mission for the platform was really to create a vibrant academic and research environment that transcends national boundaries, where we can ensure, a proactive, responsive, and proactive and ethical, high quality translational genomic research in Africa, with the focus in this case of infectious disease. There are also that are working in the space of non-communicable diseases. So, and then our goal was really to start thinking about how, was to develop that critical mass of well-trained African scientists that can use, that can be empowered, to use genomic-based tools and knowledge in order to work toward prevention and elimination of infectious diseases. And in order to do that, for instance, we thought about not only to provide short-term training for specialized skills, but also thinking about long-term training, that is training those who could be trainers of two orders. And then as such, we set up an international, master's and PhD program that, would focus on genomics, and infectious disease. And also hoping we are going to be training people how to engage communities for public health, action. So, and the research driving this program actually was focused around something that is very peculiar, that cut across most infectious diseases on the continent, that is actually identifying, the pathogens that are driving febrile illness on the continent, and that we could actually not only unravel, what we call the microbial landscape, but actually using such information as a way to provide, solutions. We're talking here in terms of diagnostics, we're talking about therapeutics, and then we're talking about vaccines. And that's really what we've tried to do over the past few years. So in bringing this, this education platform and the research platform and the research platform together, we realized that we're really meeting one of Africa's critical needs for genomic education and research. And then that really, we should look at this Venn diagram, that where the education and research cut across is really about, about genomic capacity building. That is building infrastructure, training, and also setting up diagnostic core facility, and also more importantly, actually providing, a platform, or providing, the leeway for sustainable, careers development on the continent. And ultimately, our goal is basically not just to do the research and then the academia work that we're doing, but is actually to build a next generation of African pathogen hunters who will hunt this pathogen, characterize them, and then prefer solution in Africa, and then therefore provide and giving Africa this prominent role that it actually deserves, in the community of nations, and then give Africa a voice, when people are talking about providing solution to problems. So, and then one of the very first demonstration for this really came out of the 2014, 2014 to 2016 Ebola outbreak in West Africa, where we, for the first time, actually demonstrated the transforming power of genomics in terms of, guiding outbreak response. And this was the first time this was being done. And then I know we faced a lot of challenges at the time, where we were talking about how do we use genomics and then create, you create open data sharing or open data access, in order to actually use information and make information actionable in order to guide public health response. And we're glad that, what we pioneered at the time in 2014 has become the standard. Today, we're hearing about GISED and people dropping, data in GISED, all of the countries in the world. But yet, the story is not being told completely. We pioneered that whole process in 2014 by making available the first 99 genomic sequences on the continent. Became very important because, at the time, you really did what the world did not thought that it was impossible to do. As I was saying before, we generated the first 99 genome of Ebola. We made it accessible. So, which means we pioneer open data sharing and open data access for vaccine developers, for clinical, for therapeutic developers, and then for diagnostic developers. But then what is really important is the fact that we came up with a new way and a new technology that is basically transforming, the way you could even do genomic during an outbreak. A process that could have taken about a month we actually cut down, developed new protocols, and then made it within 10 days. That is from sample collection to generating, the sequence data, and then making the data assembly, and then dropping them in NCBI. This was really 10 days, and this is something that was never seen before. So, and this is what we call transformation. We made this information available for months, and then our publication really could not come out about 6 months after we dropped the information, opened the data, to the international community. So, but what is really important I want to show here is actually the power behind genomic, where we show for the first time what is being used today as a standard. That is the case of Sierra Leone, where we show the incursion of 3 different lineages in the country, follow that over time, and then we show that, at some point around June, for instance, one single lineage has taken over and became the most transmissible lineage, and this was the first time you could use this kind of, use this kind of platform to demonstrate, the evolution, of how you could have new variants taking over in the process of an outbreak, showing that, within an environment or within a country, for instance, we had, sustained human-to-human transmission. There was no introduction of new variant from animal reservoirs, and there was no imported, it was that community transmission going on in the country. And then this information were given in real time to public health authorities. For the first time also, that actually we identify some specific, within the specific areas of the genome of those viruses, and then we actually characterize this particular variant that was associated with increased transmissibility. So, and this has become really the standard, and then, and then we really have, we feel happy that we actually set a standard in 2012. But what is really critical here that I want to show you in this slide is how, for the first time in the history of science and then in epidemiology, we're able to identify those viral genome factors or virus genome factors that actually that encourages or sustain the spread of Ebola within not only Sierra Leone but across West Africa except Nigeria. So if you look in this slide, as you see in times and then in space, you could see here how we're showing within time and space how the virus is transmitting from person to person, country to country, countries to countries, and then showing the how, and this really actually was a very first demonstration of genomic epidemiology in action, and this information, became very critical, in terms of containing this outbreak. But what we've done, as I've mentioned before, that is translating genomic into what we call point-of-care diagnostic. We started with high-fidelity sequencing. Yes, we actually, also use that to refine the PCR diagnostic, but we realized, for instance, that PCR was taking a long time. And what you need during an outbreak, are 2 factors: speed and accuracy. Speed of the process, speed of execution, and then accuracy, with what you're doing. And that's what we did here, basically by going from genomic data and translating that into what we call the very first rapid diagnostic test that could detect Ebola and confirm Ebola in 10 minutes. And this was the very first rapid diagnostic test that was approved by the WHO and then the US FDA. And then you could see that here. So the introduction of this test has, in West Africa was one of those major things that started bending or flattening the curve, because the doctors or the people could see in real time what Ebola was all about. So we actually applied the same knowledge and same skill almost in real time in Nigeria. And that's how Nigeria got in the history, the annals of medicine, and the annals of epidemiology, because this country of more than 200 million people, we actually leveraged knowledge, skill, and genomic technology in order to guide the government in its public health response. And then Nigeria actually ended up with only 20 cases and 12 deaths. But this was really genomic and epidemiology in real time, where we can identify the areas of hotspot and guide the government. So this is really showing one example how you could actually use genomic power in order to respond to an outbreak. So again, the second thing that I want to mention here is actually to show the power behind genomic and infectious disease. This is a case of a virus called Lassa fever, Lassa fever, that emerged in Nigeria, or that was described for the first time in Nigeria about 53 years ago, and then this virus was circulating in this country, and then for decades, there was no diagnostics, and then the sample used to be sent to Germany, and then often when the result of the sample come back, then 90% of the people or the cases are dead, and then these results were completely useless. In 2007, I took that challenge actually to see how we could explore and then develop diagnostics for this virus, and then within 6 months, we set up a next generation platform, by 2011, we went on to sequence viruses directly from patient samples and rodent samples, and effectively, came up with the first genomic map of the Lassa fever virus. Not only we set up the map, but we also, made some very interesting findings. One, for instance, is that this virus that was described in Nigeria just about 53 years ago, we found that that virus using the most remote common ancestors is actually about 1,060 years ago, and it emerged from present-day Nigeria. It was circulating within Nigeria years and moved out of Nigeria about 400 years ago. Yeah, what is really important about this finding is the fact that we not only created the largest genome data set for the Lassa virus, but we also translated that knowledge in developing the pan-Lassa rapid diagnostic test, and this has been very critical for the control of this disease, over time. But what are the lessons here, and then really the message here behind this genome, the whole genome sequence of the Lassa virus, is the fact that in the past 5 years, between 2015, when we published this sequence data, and now, there have been 3 major vaccine candidates that have been developed from the sequence data that we actually generated. That is showing how you could speed up a process and then find solution to a problem that has been ignored over the years. So, and then that really is very helpful in the sense that now we have 3 vaccine candidates against a disease that was neglected over decades. So this, we really were lucky to just make this publication, but also had a cell coverage. And then again, here is showing again powerful genomics that we did the first metagenomic clinical samples of pathogen, in Africa. And then this is here to show the power behind pathogen discovery. So using metagenomic as a way to go on and discover 2 novel rhabdoviruses, and then name those viruses Epomavirus 1, AKV1, and AKV2 from the area where we described. So what is critical about this, or what is important about the 2 new viruses, is the fact that they actually have a strong similarity with the Bakongo virus, a virus that was discovered in the DRC and was responsible for a viral hemorrhagic fever over there in 2012. But interestingly, in Nigeria, for instance, when we went back and then looked into those people, they all had like very high exposure to this virus. Then, Genomics for Public Health actually having demonstrated that in 2012 or 2014 with Ebola, we actually had to use that again in 2018 when Nigeria was faced with a major surge of the Lassa fever virus. There were many, public health questions that were asked, when we're dealing with new variant. It was a surge due to human transmission. You're talking about 200-fold increase within 8 weeks. So that is, you have more cases in 8 weeks than you've had in the past 2 years combined. So then this question, we actually to swing, we use genomic as a way in real time to respond to this question, and then to help the WHO and the Nigeria Center for Disease Control to address this problem. We sequence these viruses, and then we made information available. Thanks to the guys in my lab, then we demonstrated that there was no evidence of particular new strain or no evidence of increased sustained human-to-human transmission, but that this was actually, the search was due to multiple introduction of genetically independent viruses. Depending on where you were in Nigeria, you could actually see that the northern part of the virus, Nigeria has a variant of 3 or lineage 3, the west was lineage 2A, and then the east was 2B. So basically a country dealing with an outbreak in 3 dimensions, depending on where you were with the virus. So this was a shout out by WHO and then Africa City and the Nigeria Center for Disease Control for what we've done. But again, although we published this paper in New England, we realized that there was one major caveat. That caveat was actually due to the fact that we realized that the viruses were coming from rodent, and then that really helped, how to link up. We started a nationwide, rodent trapping, and then are trying to look at genetic diversity in the rodent and in human. And for the first time, you could see here in red, we were demonstrating the link between a rodent sample and an animal sample, that is showing the direct link between what happens, in animals and how this is done from animal to human. So, but when we didn't stop there, we actually adapted and updated our games and basically started using, gene editing technology, which is a Sherlock, CRISPR-based technology, to actually develop a novel kind of diagnostics for Lassa fever. So in this case, using Cas13a, and then we're able to actually come up with a diagnostic that could be done at room temperature within 5 minutes. You could get a result, and that really became very important. That is basically describing the whole process here, where you use your Cas guide RNA and then adding the nucleases and then adding a reporter, so you end up having a diagnostics like the one that I'm showing here. So, and then this really also became like a major breakthrough. I mean, there was the first demonstration of using CRISPR-Cas13a, for instance, for a disease, and then this really showing the team in my lab. I've been doing all this work, and you can see here one of my graduate students, Victoria, doing this work, and then this was really a nice publication into Nature, where Amy Maxon embedded in a lab for a week actually to see this work being done from the beginning to the end of the process. So this other slide is really actually to show also real-time genomic analysis, when you want to talk about how you can use this actually to uncover outbreaks. So in this case, this is a case where we use metagenomic to uncover an outbreak of yellow fever somewhere in the southern part of Nigeria. So this was in 2018. We had, within a week, we had 179 school children that died in the community. All the tests, proved negative. Lassa, Ebola, and everything proved negative. The Nigeria Center for Disease Control did not understand what was going on. They sent us 22 samples, and then we went in there with very powerful metagenomic approaches, and we were able to identify a new lineage. So here, new strain, you could see them clustered in red, of yellow fever circulating in Nigeria. But what is important about this finding is the fact that this new strain of Lassa fever, yellow fever, that was circulating in Nigeria and was negative for all the tests, was due to the fact that it was diverse and it was very different from the ones that have been circulating in the country in the past 94 years. So really showing how if you embed, like this kind of powerful technology in the silos, laboratory silos method that are existing today, you can make a big difference. And then this is, again, a typical case also whereby in Nigeria, this paper came out about on Monday this week, where we're able to use, next generation sequencing to identify an extensively drug-resistant tuberculosis in Nigeria and West Africa. And this is the very first time, that this description is made in West Africa, whereby we really use genomic data actually to prove that we're dealing with a strain of TB that is not responding to any of the drugs that are available. So really showing again, behind the detection of this technology, so we can actually show clearly. So the case of HIV, for instance, is another case here, for instance, where we use, genomics as a way. I mean, one of the first, whole genome sequence project being done in Africa, where we were actually not just looking at the envelope or the GAG or the pole gene of HIV to characterize the HIV-1, but really looking at whole genome sequence. And that has really helped a lot, because it not only just helping us to understand exactly the genome, the HIV diversity, but also understanding actually spread and an importation of diseases. In this case, for instance, we've been able to show how the viruses have been brought into Nigeria from neighboring countries by just using genomic sequences to track, some of these viruses migrating or moving or migrating through human from country to countries. And that becomes very important as a way, to do this. So again, just along the line, we have a very powerful bioinformatics team that is developing new toolkits. For instance, like here, this is something that is published, will be published on Monday in PeerJ, which is a bioinformatics journal. This is basically a viral genomic, assembly platform that my team has just assembled, and then you don't need a server, just download it on your system or your laptop, and then you can actually do all of the bioinformatics analysis, just put in your data, and then you can get the output. Now, let's fast forward to COVID, and then COVID, as really struck Africa, a bit early in 2020. And then the very first country that actually had COVID, one of the first countries was Nigeria. And then when we got the samples in Nigeria, when the first suspected case arrived in Nigeria, then the Nigeria Center for Disease Control had to just get the sample down to us, to our lab, basically knowing exactly the capacity that is available and then the level of automation that we have here. So you could see here, for instance, the automation and then the platforms, and then you could see here the next 6,000 still remain the only piece in Africa. So, but then we basically just pushed this, and then within 48 hours, we're able to actually first, come up with the first SARS-CoV-2 genome sequence, in Africa. But then this information became very critical because we knew that definitely this information, I mean, this virus was imported and it wasn't circulating in Africa. And then together with other colleagues, you know, along, I mean, in the next few months that followed, we were able to show, for instance, that there was about 761 importation of the virus within the continent. And then these were mostly, you know, this data was shared with the Africa Center for Disease Control, and the reason why Africa shut down during the first and second wave at the same time, actually preventing the continent to go in, I mean, to spiral down like other part of the world. But then we use this information not only to inform public health policy, but also to set up a surveillance system within Nigeria. So, but then this slide is a bit outdated. I think over the years, the past over 1 year, we've done over 1,000 sequences. I mean, we've covered more than 2/3 of the Nigerian states. But then in the case of Nigeria, we know exactly at each level where, I mean, in each state, what are the variants circulating, and these informations are given almost in real time to the National Center for Disease Control and the Presidential Task Force so that they can use in order to respond to the pandemic. So in this case, for instance, this is an X-strain analysis. So then, and this information is shared, you know, with the Presidential Task Force for policy making. So a lot of the decisions that they're making countries are based on this data. So which means that the information or the policies are evidence driven. We know the different lineages circulating in Nigeria. In the last count, there were over 45 different lineages. So we know exactly during the lockdown, even in Nigeria, we know, you know, if you look at this year between the different states, you could see variant moving from state to from state to state. And that's really was very helpful at the time to help the government in order to devise strategies, you know, to prevent, you know, people to move, because we realized at some point, despite the lockdown, things were moving. So, and one thing I want to highlight here is the fact that, you know, the genome sequence data that we generated were not just used for public health, you know, response, but we also translated that into point of care diagnostics here. This is all the SHINE technology that we've just developed. This is also done at room temperature. And then you have the format that could be done on strips that can be done in the most remote part of the world, but we also have the plate format that can be done, I mean, to increase the throughput, where we could do 96 samples at a time. And then this kind of a very first test that we did in Nigeria. I mean, at the time in our lab, like 500 first tests, and I was very happy. I mean, happy to see that. But then what is very important is the fact that this was within 2 weeks of the outbreak, and that we were able to come up with this new technology out there. But what is important in the fact is the fact that we know now that within a week, if we have an outbreak, we should be able to come up with a solution and then help, you know, I mean, there's the help with this. But interestingly, we are also now multiplexing, you know, this in our lab, and basically bringing together all of this technology, I mean, all of this pathogen under one single diagnostic chip, and whereby we could actually go, I mean, test up to over 160 different pathogens in one single test and assay. We've also translated this into coming up with like a vaccine candidate for some of you that know, and that has gone through preclinical trials, and it will moving on to clinical trials. But the story behind the vaccine and the reason why we're not funded to compete with others, because it's again the same messenger RNA technology platform that we developed and designed here, but we couldn't move it forward because of science politics and also lack of support from African government. So I really would not want to bother you with all of this, but then this is a subject to a different conversation. But one thing that we basically do want to say here is that actually we basically design a vaccine candidate that is mostly adapted for the continent. We do not need temperature change. We do not need, I mean, a needle. You know, we definitely is a needle free because we understand exactly the context in which we evolve in. So, and then again, to say that over time, we've not just worked only with Nigeria, we've also supported many African countries. And as I can see here, identifying variants of concern and variants of interest. So we've basically done a lot in supporting not only Nigeria, but many African countries. And then this is here, for instance, whereby we've been able to help about 14 different African countries to sequence, I mean, their samples, and that has generated over 2,500 different genome sequence of SARS-CoV-2. Training also the capacity in those different countries has helped. And then in this case, for instance, you know, we've generated over 2,500. We've also trained a critical mass of people since 2014, over 1,000. But what is very impactful is the fact that the case of countries like Ghana, for instance, Brigadu Noguchi is someone that we train, and then today is is one of the leaders in West Africa in, performing genomic sequences. And then we also had the case of Rwanda that we actually trained, Rwandese scientists, and then we went back to their countries, and within a week they started generating, sequences on their own. And that has been very, very helpful because we actually building that capacity on the continent in a way that was unprecedented. But what we've done is that what we've been doing is not just to train, but we're also following that through with what we call infrastructure. We're setting up sequence platform across the continent, and that really has been very helpful in terms of helping Africa to respond. Finally, what I'm going to close on is actually to talk about this new platform called Sentinel, a new project called Sentinel that is really built around making Africa lead pandemic preemption and response, and build on genomic technology and then what I call information technology as well, and information sciences. It's really built around three major pillars. The first is to detect known and unknown infection almost in real time, and then pass that information to public health authorities, locally, nationally, and then internationally. That brings us to the second pillar called Connect. And then the final one is empower. How do we empower the public health community, including, the national public health and then the frontline healthcare workers, in order to harness these tools and technologies in order to respond to the pandemic on time? You all agree with me that if we have this kind of platform set up before, I'm sure this would have been detected, or the SARS-CoV-2 would have been detected much more earlier in Wuhan or in China. This information would have been shared globally, and then the world would have responded more quickly. So this would have put us in a very different position compared to where we are now. So our goal is actually to use this system where we build around what we call the hubs of excellence here in Orange, and then the spokes, which are the satellite sides of the hub, will now work with the communities. So, and then this is a hub and spoke approach that actually connect the hub and the spokes and then the satellite communities, and so that we can have a global system, or a system that can enable us to respond almost in real time. And then, once we have created a system that will connect all of them, and then eventually you will have to empower them by just ensuring that everybody becomes a sentinel, and then we'll start looking. We've started pioneering this system in Nigeria, and then we're actually going to be establishing the same system in Senegal, Sierra Leone, and then the DRC. Yes, we are basically going down to a point whereby we think that we can work with the public health authorities, and then we can actually set up this platform whereby we can actually refine the high functioning technology, working with the communities in order to come up with a situation whereby we can empower, Africa's scientific capability, and also and set up really what we call independent surveillance and outbreak response on the continent. And this model can be replicated across the world. I really want to end by acknowledging, not the team working with me, but also a lot of people funding all of the work that I talked about. And I also want to thank you again, and then for listening. Thank you.
Thank you so much, Christian, for a wonderful presentation. I've never felt tired listening to your talks. And I wish we had more time for Q&A and more insights from you, because we do know what you have actually done in Africa and how that will impact how we do pathogen genomics in the continent and beyond, particularly since this is at the core of my ambition and aspiration as a scientist. So thank you so much, Christian. I hope we'll find some time for Q&A. I kindly request all the participants and attendees to send their Q&A in the chat box. And if we have some time, we'll discuss them at the end. I would wish to take this opportunity to introduce my colleague, who is also a board member of the African Society of Human Genetics here in Tanzania, Dr. Dan Maeda, a lecturer at the University of Dar es Salaam, and he has chipped in at a short notes to co-chair with me, and Dan will introduce our next presenter. So welcome, Dan.
Okay. Afternoon, everyone, or morning, depends on where you're from. The next speaker is Segun Fatumo. Segun Fatumo is an assistant professor of genetic epidemiology and bioinformatics at MRC UVRI. That's the Ugandan Virus Research Institute in Uganda, and also at the London School of Hygiene and Tropical Medicine. He leads the Africa Computational Genomics Research Group. His group's expertise include, but not limited to, GWAS, meta-analysis, multivariate GWAS, genetic risk score, fine mapping, Mendelian randomization studies, and drug targets discovery. He was recently awarded a prestigious Wellcome Trust International Intermediate Fellowship to lead an innovative research program at the MRC UVRI and LSHTM. His research focuses largely on the genetic impact of non-communicable diseases. In Africa mainly, he's actively involved in capacity building across Africa, including teaching and/or postgraduate supervision at Makerere in Uganda, the University of Science, Technology and Technologies of Bamako in Mali, and Covenant University, Nigeria. He's also part of the H3 Africa Bioinformatics Network, H3 Bionet, heading the node at the Center for Genomics Research and Innovation in the National Biotechnology Development Agency in Abuja, Nigeria. He's an associate fellow of the Higher Education Academy in the UK and teaches MSc programs at LSHTM and Queen Mary University of London. Dr. Fatumo co-leads the Nigerian Non-Communicable Disease Genetic Heritage Study Consortium and co-directs the Nigerian Cardiometabolic Genomic Study, which is a concerted effort to see over 100,000 Nigerians participate in the eponymous study, which will seek to understand the genetic basis of the highly prevalent non-communicable diseases in Nigeria, such as chronic kidney failure, diabetes, obesity, among others. In today's talk, Dr. Fatuma will talk to us about the genetics of cardiovascular diseases. I'm sure it will be very interesting. So, Dr. Fatuma, if you're ready, please begin.
Good day, everybody. Good morning, good afternoon, and good evening, depending on where you are joining today. I would like to start by thanking the organizer for this, for inviting me to make this presentation today. My name is Segun Fatumo. I'm currently an assistant professor at the MRC UVRI and LSHTM in Uganda. So I lead a research group that is called the African Computational Genomics, TSHG. So today I'll be making this presentation that I've titled Genetic of Cardiovascular and Related Diseases in Africa. If you have any question that time does not allow me to talk about or to comment about, please feel free to send me an email. My email is on this slide, or you feel free to just contact me on Twitter. I'm very present on Twitter. So as a way of introduction and background, you may already know that cardiovascular disease, CVD, is a general term that is used to describe the disease of the heart and blood vessel. An example include coronary heart disease, heart attack, and stroke. An estimated 18.6 million people die of CVD every year. This represents 33% of all global deaths, making CVD the world #1 killer disease. So CVD can be caused by a combination of a genetic predisposition and traditional risk factors, such as high blood pressure, high cholesterol, obesity, diabetes, and kidney disease. I think that understanding the risk factor is key to finding some kind of solution to the cure for CVD. Unfortunately, the burden of CVD and its risk factor is increasing in Africa, with expected deaths projected to double in 2030, according to a report by WHO. And given the central importance of Africa to our human origin and disease sustainability, there's a clear scientific and public health need to develop large-scale efforts in examining this disease across diverse populations within Africa. So like you're all aware, genome from Africa ancestry individual are scarce. Most of the time, when I make these statements, I like to emphasize that human genome data from continental Africa is very, very scarce. particularly when you compare to what we have from the European and other populations. Like you have already heard from some previous presentation, less than 2 to 3 of human genome analyzed so far have been from African ancestry individuals, despite the fact that Africa contains more genetic diversity than any other continent. Again, I always like to emphasize that those 2 to 3% African genome analyzed are mainly genomes of African American individuals. For example, in the past few years, there have been significant advancements in identifying genetic factors that contribute to CVD using approach called GWAS. And there have been an association of from 100 to 1,000 loci that are known to associate with CVD, but little from Africa. So while we aim to address the genomic imbalance by various efforts in the continent and globally, we as Africans must learn to maximize the use of our rich African data set in the continent. In this presentation, my aim is to demonstrate to you how we, the African computational genomes, are maximizing the genomic resources in Uganda for novel genetic discovery, polygenic prediction by leveraging strategic regional and global collaboration on genetics of cardiovascular disease, while we also build capacity building and train next generation scientists in Africa. So in order to discover genetic variants that contribute to CVD and its risk factor, my group is using approaches such as GWAS, fine mapping, polygenic risk score, and Mendelian randomization study to infer association, causality, and as well as prediction. So using GWAS, for example, we are interested in identifying what genetic variants are associated with a range of CVD and its risk factor. Once we identify such association, we refine them. We refine those associations as known and null loci. We also use an approach called PRS to estimate individual genetic liability to a disease. A PRS is we calculate the sum of risk allele that an individual has. We weigh them by the effect size as estimates using the GWAS on the phenotype. So ultimately for PRS, we are interested that we may be able to help identify subgroup of individuals in Africa who are at high risk of CVD. So very important approach we also use is called Mendelian randomization approaches. Like you may already be aware, observation studies have shown that there have been an association between CVD and a range of risk factors that I've mentioned, like type 2 diabetes, kidney disease, hypertension, high cholesterol, and the rest of them. So this kind of studies can be limited by failure to adequately control for confounders. So while we use Mendelian randomization study, it will help us to assess the causal relationship between a range of CVD and its risk factors. So first, in Uganda, we set up a genetic epidemiology study using the cohort called General Population Cohort, GPC. GPC is a population-based cohort of about 22,000 people that live in a rural community in Uganda. So this cohort was originally set up in 1989 to examine prevalence of HIV in Uganda population. From around 2010 and 2011, the research activities at CPG was broadened to include epidemiology and genetic epidemiology of non-communicable disease. So every year, our team at the MRC go to the community and recruit people, also measure a range of indices. So we always start by sensitizing the local community about the study activities. We seek their permission before we go into the community. In the last few years, we have a group that is called a CAB, Community Advisory Board. So this group includes people in the community, very important people in the community, like pastors, like imam, like cultural leader, group leader, as part of our Community Advisory Board, and as well as a professional at the MRC UVRI. So this group, they give us guidance on how to do our research and how to enter into the community. So in this particular study, 5,000 individuals were recruited for genetic epidemiology studies. Another 2,000 individuals were recruited for whole genome sequencing. Yeah, so we went ahead, we collaborated with similar studies in Africa. We collaborated with the ADAM study. ADAM is the African American Diabetic Mellitus. So this cohort is led by Professor Charles Rotimi at the NIH. And this cohort has an individual from Nigeria and Kenya and Ghana. We also incorporated data from the Durban Diabetes Study. That's a cohort from South Africa led by Aisha Motala. And altogether, we have pull all this data together and have a total of about 14,700 individuals across Africa. So as you can see on this slide, all the study measure a range of CVD risk factors, including HbA1c, which is an indicator for type 2 diabetes. We have a lipid trait, we have blood pressure, we have full blood count, and other traits. So in order to demonstrate the value of African genomes, we published this work in Cell. The project represents one of the largest collection of GWAS and whole genome sequencing in Africa. So as you expect, we found a high level of genetic diversity in the Uganda population than you will see, for example, in the European population. We found specifically 28.7% of SNPs discovered in Uganda whole genome sequencing data have never been found before. This was about 9.5 million novel variants, representing about a quarter of genetic variants identified in Uganda population. So this slide shows the list of novel and distinct genetic associations that were found to be associated with various cardiometabolic traits and disease. So as you can see on the last two columns, this column and this column, with the heading minor allelic frequency for Africa and minor allelic frequency for European. You will have noticed that here we have mainly a 0 minor allelic frequency, meaning that some of these SNPs are not available in the European population. And this again also emphasizes the need to study African population. And this SNP, which are monomorphic in European population, are found to be associated with a range of cardiometabolic traits in African population. So this is very, very important. So our study also leveraged on the lower linkage disequilibrium in African data set. The fine mapping from our analysis resulted into a narrow credible interval for most of the traits. We were able to map 16 out of 41 distinct loci to a single SNP in the credible interval. We also resolved the previously identified association with HbA1c. So I think fine mapping is an opportunity for Africa. It's an opportunity, particularly when we use the African data set. So the richness of the Uganda genome resource also offers us the opportunity to study the extent to which genetic differences encode differences in traits. And I'll give an example of height, and I'll also give an example of LDL. So we assess the heritability for all the 34 cardiometabolic traits using mixed model approaches that also model environmental correlation. So we found that height, for example, is less genetically determined in Uganda relative to what you will see in European studies. So the narrow sense heritability for height was 49% in Ugandans, compared to an estimate of about 70 to 80% in the European population. So one reason for this might be environmental factors such as nutrients in Uganda. So in contrast, the heritability estimates of LDL cholesterol were significantly higher in the Uganda population, and that was about 54% compared to 20 to 43% in the European population. So on the whole, our finding highlight the usefulness of examining genetically diverse population within Africa and also other populations. So from there, we have now recently extended our collaboration to individual data of African in diaspora. We are collaborating with colleagues on the use of the Millennium Veteran Program, MVP, which also has a range of cardiovascular disease measure. And this cohort has about 57,000 African ancestry individuals. We are also collaborating with data of about 7,000 Black participants in the UK Biobank. So recently, we aggregate all the African ancestry data sets, making a total of about 78,000 individuals of African ancestry. So in this study, we identify more than 400 genetic variants that reach genome-wide significance for diastolic and systolic blood pressure. Of course, most of these genetic associations were driven by the MVP dataset. So Brenda Idosen, who was an MSc bioinformatics student, performed meta-analysis and multi-trait analysis at GWAS to infer a better approach for gene discovery. So in this work from Brenda, so we show that leveraging collaboration between traits like systolic and diastolic blood pressure, it can improve power of genetic association to identify a marker that have not been previously associated. You can see from this Venn diagram, you see using multi-trait, you have an extra gene discovery that were not found using meta-analysis. So similarly, using the same data set and also using a similar method, but this time around, different traits, lipid traits. So Toure, who is from Mali and also doing MSc bioinformatics, identified 6 novel loci using a univariate method and 10 novel loci using multi-trait method across all the 4 lipid traits. So we leverage the shared information between the traits to perform multi-trait file mapping using a tool that is currently being developed called FlashFM. So FlashFM is a Bayesian file mapping method that explores the shared information between those traits, like lipid traits, LDL, HDL, and total cholesterol. So this figure shows comparison of credible set size across loci with multiple association with FlashFM when plotted against a very popular tool called FileMap. So both Brenda and Toure are currently writing up this work for publication. Again, using the same data sets, we assess the performance of lipid trait PRS that were derived from summary statistics from European population and African American, multi-ancestry individual, and as well as a multi-trait approach to understand the transferability of PRS from different populations to African population. So the S-axis, for example, show different ancestry. Like I've mentioned, you see here is blue, here is the African population. You see the MAA here is the multivariate where you jointly assess the different lipid traits. So here we show that African American derived PRS were more predictive compared to multi-ancestry and other PRS. And this is very important because current publication and the correct concept is that using multi-ancestry derived PRS is enough to predict into any population. So notably also from our study, we see that using the multivariate derived African American PRS was even more predictive than univariate approach of multi-ancestry and as well as the European population. Here you see the African population predicting far better. You see the multi-trait predicting much better before the multi-ethnic population, which is currently being said to predict better in all populations. So our result is a total divergence from the PRS plot that you are familiar with. Many people are familiar with this plot from a paper that was published by our friend Alicia Martins, showing a poor predictivity into African population. From our study, you will see that African predicting better because we are using an appropriate data set. So again, our study suggests that improved representation of African individuals will ensure generalization of findings for precision medicine. So in another study from our group, which this work, for example, currently is under review, we show that the African-American derived PRS was more predictive of type 2 diabetes compared to multi-ethnic derived score. So this finding, it was from one of the cohorts from the Zulu cohort in South Africa. It was also replicated in the Adam cohort from Nigeria, Ghana, and Kenya. So again, so this seems to be a contradiction to previous study, which suggests that PRS based on data from multi-ancestry population improve prediction for all population. So most important from this study, we found that the participants in the 10th decile of the PRS had more than 3-fold greater risk of developing diabetes, and they were diagnosed 2.6 years earlier compared to those in the first decile. We are now expanding this type 2 diabetes study in the work of Chisholm Shuremekun doing a PhD at Makerere University in Uganda. So Chisholm is looking to create the African ancestry genomic architecture of type 2 diabetes. So she aims to include the evaluation of transferability of PRS in different regions of Africa. For example, we want to know how to PRS transfer between the West, East, and the Southern Africa. So in a pilot work, she meta-analyzed type 2 diabetes viewers in 3 studies, the Adam cohort, the South African Zulu cohort, and MVP, and found 4 distinct associations. So she's now exploring how best to find map the novel loci and as well as the no loci of type 2 diabetes. So using the results from meta-analysis of lipid traits and type 2 diabetes from both Ture, Chisholm, and Brenda, and other people from the group. So recently, we investigated the causal effects of type 2 diabetes liability and lipid traits on the risk of stroke in African ancestry individuals. We compared this to the estimates that have been obtained from the European ancestry individuals. Like you can see on this slide, so we find that higher genetic proxies type 2 diabetes liability and LDL, total cholesterol, and lower genetic proxy HDL were associated with increased risk of stroke in African ancestry individuals. So we also found that there's no evidence of differences in the estimates. So when performing analysis in the European ancestry and as well as African ancestry individuals. So this work involving many members from our group has now been published in a journal that discusses stroke. So again, our analysis here support a causal effect of type 2 diabetes and lipid traits on stroke in African ancestry individual, which is very similar to what you also see in European ancestry individuals. So considering the high burdens of kidney disease in Africa and Uganda, we're now leveraging the use of a biorepository to measure serum creatinine in Uganda and as well as other kidney biomarker in Uganda. So these resources are very, very important, particularly for sustaining genomic study in underrepresented population like ours. So recently we performed GWAS in 3,000 individuals in Uganda. We identified and validated 2 EGFR loci at HPB and as well as GATM. So as GATM locus, the association signal in Uganda was distinct from previously reported signal and this locus. At the HPB, the association signal has been previously reported, but the LISNP at the HPB loci accounted for 88% of posterior probability of causality after fine mapping. So this work again was published recently and also again represent the first GWAS of kidney function in Africa. Our findings further highlight the importance of diverse ancestry for uncovering novel association. Even now, so larger continental African meta-analysis are now needed to gain further insight on the genetic architecture of kidney function in African populations. So this is from Richard Mayanga, who is a second-year PhD student at Makerere University in Uganda. Richard is now expanding on our kidney function GWAS work. Richard is focusing mainly on aggregating genomic data set of kidney function from continental Africa. So Richard's work involved expanding the UGR, that's the Ugandan Genome Resource, from that 3,000 individual that I showed previously, and she's now trying to expand that by meta-analyzing them with other important data sets from Africa, such as the ADAM data set, led by Charles Rotimi, the DDS from Aisha Motola, AWGEN data set from Michelle Ramsey, and Meru data set from Mia Cramping. So like you can see here, so the Meru data set is a Brownian cohort for genomic study. So we are currently genotyping up to 8,000 samples on H3 Africa array. So Richard will be using this resource to build capacity in conducting genotype calling, doing quality control, doing genotype imputation, and as well as doing GWAS. So in a pilot study that was done by Richard recently, so Richard meta-analyzed UGR and ADAM cohorts. We found a novel loci at chromosome 1, which was implicated in a gene called PDZK1. So using GTES and other gene expression resources, so we found that PDZK1, a protein coding gene, is highly expressed in kidney and liver. So we think this is a very interesting discovery for kidney function, which we're now presenting here for the first time. So Richard is going to be working more on this to investigate this further. So Richard is aiming to publish this work after aggregating more data set from AWGEN, from DDS, and as well as Meru cohorts for his PhD work. So as part of our investigation to understand kidney function in African population, so Miriam, who is a Richard Fellow in our group, is working to determine the frequency of two ApoL1 risk allele, G1 and G2 in the Ugandan population, and also trying to investigate the association with kidney function, blood pressure, and bilirubin level. So this slide, for example, shows high frequency of 1 or 2 copies of ApoL1 risk allele in West Africa, in Nigeria, Gambia, Sierra Leone, than you will see in the South Africa and West Africa. You will see here the blue color showing no risk allele, and so yellow color showing 1 or 2 copy of the risk allele. And you can see a pattern of high frequency of 1 or 2 copy of risk allele in the West African population, and you will see a lower frequency in the East African population and South African population. So, and again, this is also consistent with prevalence of kidney disease in different regions. So whenever you see high prevalence of kidney in African population, we also see high frequency of 1 or 2 copy of the risk allele. We think this is a very good pattern, and Miriam is looking to write this up after further analysis. So in our recent published work on GWAS of kidney function in the Ugandan population, so we reported that multi-ethnic based PRS do not predict well in the Ugandan population. So Chris Kintu, another PhD student at Makerere University, is developing a PRS score for kidney function from diverse population, and he's trying to evaluate the transferability of a kidney function score in multiple ethnic groups in Africa. So this genetic score, when developed, can be used as instruments to assess whether cardiometabolic traits and disease are causally related to the risk of chronic kidney disease in Africa. And Okwe, who is a postdoc in our group, is leading a bidirectional 2-sample Mendelian randomization study using EGFR as both exposure and outcome to investigate the causal relationship between each cardiometabolic trait and kidney function. So just to summarize everything I've been saying, so most large-scale study of genetic of CVD and its risk factor, they have been dominated by participants of European ancestry. So therefore, African ancestry population currently lag behind. So in our research group at the TSHG, so while we aim and we are also looking for a way to address the genomic imbalance, so we are using approaches such as GWAS, file mapping, polygenic risk score, and metadata attribution study for gene discovery, for polygenic prediction, and also investigating the causal effects of modifiable risk factor on cardiovascular disease in Africa. So our ultimate aim is that we might be able to find insights that will guide the prevention and treatment of CVD in Africa. To conclude, I would like to thank all members of our group who have done most of the work that I've presented today. We have Okpe, who is a postdoc that is doing very well. We have 4 PhD students, 3 of them are registered at Macquarie University. Sarah Silva registered at the London School of Hygiene and Medicine in the UK. We have a PhD student and also a research fellow that have been making a lot of contribution. I also like to acknowledge our friend, our partner, that are also contributing to our study in the group. And most importantly, we have been very, very fortunate to collaborate with a range of leading research groups in Africa and globally. Particularly, I would like to acknowledge Dr. Charles Rutimi's group in NIH, Aisha Matala in South Africa, Michelle Ramsey, Mia Crampin in Malawi, and others. So my group has also received a lot of mentorship from these people, including Moffat Urenda at the MRC, UVRA in Uganda, Andrew Morris in Manchester, Monica Woody and Derek Widman at the University of South Florida, and as well as Stephen Borges at the University of Cambridge. So finally, I would like to acknowledge the contribution of the Consortium, the East Africa Bioinformatics Network, PTS's team at the East Africa, CKD Gen Consortium, and the Cogen Kidney Consortium. So thank you very much, everybody. Just before I go, I would like to say that we are now looking to expand our group and looking for a staff scientist who have experience in doing GWAS, file mapping, PRS, and metadata administration. We are looking for somebody that can be based in Uganda or Malawi, or somebody that can be based at home in their pajamas and do the work. So if you are interested, look for somebody that can start either in December or January next year. Please feel free to contact me. Thank you very much, everybody. God bless. Bye-bye. Good day, everybody. Good morning, good afternoon, and good evening, depending on where you are joining today. I would like to start by thanking the organizer for this, for inviting me to make this presentation today. My name is Segun Fatumo. I'm currently an assistant professor. Hello. Yes, I can see one of them. Okay. Okay, thank you. So thank you, everybody, for listening to my presentation. I can see one question from here from Dr. Makani, Julie Makani. Asked a very good question. In the group, we are very open to collaboration, and we are open to collaboration within a project within the East Africa. So currently, I'm part of the PTSD project in East Africa. I'm also fully part of the East African Bioinformatics Network. The project I've described today, particularly the project on the use of genetic for CKD, is a very important project that needs a lot of collaboration. We are now working with project with a cohort in South Africa, that's DDS. We are also establishing a collaboration with Professor Michel Ramsey. But we are also very mindful to respect that every project within Africa have the opportunity to publish their workforce before we are able to aggregate all that data together. So we are collaborating with within Asia Africa. I'm also aware that there's a project called Asia Africa Kidney Network. We're also interested in collaborating with them. We are just waiting for them to advance their work and also possibly publish some of their work before we pull together all the data sets. So that's a very good, that's very important because GWAS particularly is key when you have large data set. We are aware of that. We are also aware that other people need to publish their work before we can aggregate all this data together. I can also see a question from Professor Michel Ramsey. So Michel is asking about the capacity development through the PhD student and postdoc is wonderful. So thank you very much, Michel. I think that's one of the things that will help to sustain genomic study in Africa is strategic collaboration and capacity building. So in our group, like I've mentioned, we are trying to do that, supporting postdoc and also supporting PhD students. Like I mentioned, currently we have 4 PhD students. We have 1 postdoc. I'm also looking to also engage another postdoc by the end of the year. These are very important if we aim to sustain genomic study in Africa. Thank you, Michel.
Got lost. I hope I'm back.
Yes, you're back.
Okay, thank you so much. We had 2 questions which you helped to address. Christian, we also have a question for you from Michel Ramsey. And that was, Christian, can the assay for multiple pathogens be done at the bedside, or it has to be done in the lab? And in connection to that, Christian, it will be great if you can also enlighten us, how are we going ahead with the pathogen genomics in this context where most of the funding is actually not domestic? So you could probably couple that with Michel's question.
Yeah, the multiplex for now, we are doing it in the lab. And Michel, as you know, we have to do real developmental research at this point. But the idea is actually to multiplex them as point-of-care diagnostics. We've done a few point-of-care diagnostics, and then as of now, we know we can have like Ebola, Lassa fever, and then malaria on one single strip. But I mean, how to multiplex a few hundreds of this into one single strip is going to be a bit challenging. But we can do up to 160 on the single platform but that is basically just picking up all the specific DNA and RNAs in one assay. That is still being done in the lab, but we're hoping that, you know, technologically, as we're moving forward, we should be able to actually just couple more into point-of-care diagnostics. But I think that's an answer to that. Because I think the goal for us is not just to do diagnostic for the purpose of putting them in some lab. As you see, as a Sentinel network, we really want to make everything as point-of-care diagnostics. I think, yeah, it is challenging actually at this point, you know, to think about how do we want to move forward as pathogen genomics or even human genomics, because most of the funding is coming from outside. And I think for me, it's an opportunity here to call on the African leadership and African government to put their money where their brain and their mouth is, because it's important, you know, to invest, because the future of Africa is intricately related to investment into research and development. Africans should stop looking at research R&D as a luxury. It's not a luxury. I think it is something that we have to pay attention to. If there's anything to learn from this pandemic, is actually the vulnerability of Africa and the high reliance of Africa on outside. And we see what it has cost us, because today we have no vaccine, we have nothing, and simply because we have refused to invest what we're supposed to do. it's not because Africa lacks the resources. We do have the resources, but unfortunately, we are really not investing or prioritizing research and development. Hopefully, we can learn from this pandemic, and hopefully we can start putting the resources where our brain is, so that we can actually change the narrative and then change the dynamic of the situation on the ground.
Okay, so I've been asked to make a comment about one question raised by Mr. Ramsey about collaboration within Africa, particularly mentioned in 54 Gene. Maybe you are aware that I'm fully part of the public-private partnership with 54 Gene. So I co-direct one of the projects that is called NICGEN that aims to recruit 100,000 individuals in Nigeria. That has been a very successful project so far. In the last 1 year, on that one project, we have recruited more than 20,000 individuals in Nigeria, also with a very deep phenotyping, a range of cardiovascular disease. And there's also another project that have recruited more than maybe 60,000 individuals in Nigeria. That's an excellent project. I think in Africa, again, to sustain a genomic study, we have to look beyond funding from NIH and Wellcome Trust. I think we have to look also into partnership with private organization, biotechnology company, and all that, which is what we are doing with 4G. So I think a conference like this will give us opportunity to leverage collaboration on the use of that data. One thing that has been lingering so much in Africa is many people just tend to keep to their data, and the data will not bring a lot of results because they are very small data. But when we are able to aggregate all this data together, we'll be able to compete very well with other projects, other large-scale projects globally.
And I wish to take this opportunity to thank you so much for your presentations and all the great stuff you shared. And I wish to thank the attendees for taking time to be with us. This has been very exciting, but I think the time has not been enough. So thank you so much. And we'll be now joining other sessions. Those who might want to stay can continue the Q&A. So thank you so much and see you next time.